Hello everyone, before we start with our official test review, I want you to take out your test review sheet as well as the 4 notes that goes along with this test so that you can make some marks on it. What I want you to do is, you don't have to fill out the entire test review sheet. However, when we're going over these questions, I want you to think about whether you can answer these questions without looking at the answers or finishing that part of the video. And if you cannot answer those questions, you should make a mark next to it so that you can write down some notes for those questions so that you can go back and test yourself again. At the end of your test review packet, there is a page with all the questions without the spaces and for the answers, so you can use those to test yourself again. Any question that's on the test review sheet is fair game for the test, so make sure you can answer all the questions so that you're completely ready for Monday's test. So we'll first start with the last part of evolution. First question is, what is adaptive radiation? How many species do you start before adaptive radiation happens? Basically, how adaptive radiation works. So if you go to our lecture slide, there are 2 slides on adaptive radiation. Adaptive radiation is a single species or a small group of species evolve over a relatively short time into several different species that are adapted to different environments. So the example that you can remember is the finches example. At the beginning, those finches came from the mainland, and there was only 1 species of finch. So if you look at this picture, we started with 1 species of finch, and those finches went to different islands with different environments. So the key here is that different places that these species went to had different environment that they have to adapt to. And the result of this adaptation to the different environment is that you end up with different species of finches or different species of whatever, right? So make sure you understand that adaptive radiation is when you start with 1 species. Write this down, start with 1 species, and then you end up with many species. And how exactly does that happen? It's because the environment of the new places have different environments that the species have to adapt to. Okay, so that is how the environment affects adaptive radiation. The next one is, what is the Heterotroph Hypothesis? So if you just look at the name itself, Heterotroph Hypothesis, it's a hypothesis and has something to do with heterotroph. So what happened? If you look at this picture right here, what it's saying that is that the first organisms on Earth were anaerobic heterotrophs. Okay, I'm not going to write this down. Anaerobic heterotrophs, you should know this. The reason why it's anaerobic is because at the beginning of life on Earth, there was not that much oxygen in the air. So the organisms that are living has to be anaerobic. They're heterotrophs because they have not yet developed the ability to go through photosynthesis. So that leads to our next topic. What is the theory of endosymbiosis? The theory of endosymbiosis explains how organisms were able to finally do photosynthesis. And because of the ability of doing photosynthesis, after photosynthesis, the photosynthetic organisms, the autotrophs, produced all those oxygen that we're breathing in right now. So thanks to plants, we are able to live. So heterotroph hypothesis is right here. The theory of endosymbiosis is where you start with a primitive eukaryote, and then there's also a prokaryote. So we call it a primitive eukaryote because if you look at this picture, we're already starting to form a nucleus. So if something has a nucleus, that's a eukaryote, right? But we call it a primitive eukaryote because it hasn't gained all the functions, all of the organelles that eukaryotes nowadays have. So for example, at the time, there was no mitochondria within a eukaryote, and there was also no chloroplast within a eukaryote. So where did they get those? Those pre-mitochondria, pre-chloroplast organism were actually prokaryotic cells, and this primitive eukaryotic cell engulfs the prokaryote, and then eventually this prokaryote became a part of the early eukaryotic cell, and then both the bacteria, both the prokaryote and the eukaryote benefit from this arrangement, and eventually this prokaryotic cell, which is the previous form of mitochondria or chloroplast, became mitochondrial chloroplast and is no longer a bacteria. So we call this the theory of endosymbiosis. So you know, symbiosis or symbiotic relationship is a relationship between two organisms where both organisms benefit. So in this case, both the prokaryote and the eukaryote benefit from this relationship at the beginning. So we call it this endosymbiosis because this prokaryote had to be engulfed. And the example is mitochondria and chloroplast. The prokaryote that was engulfed eventually became the mitochondria and the chloroplast. So make sure you know these examples. If I were to describe something like this paragraph to you and ask you, what is this? You need to know that it is the theory of endosymbiosis. Or I can ask you, what is the theory of endosymbiosis? And you should be able to explain this to me. So one good way to help you practice is take your roommate, or if you live at the dorm, or your parents if you live at home, or your brothers, sisters, whatever. Explain these things back to them just like how I'm explaining it to you. If you can teach other people, or even just a stuffed animal, whatever, your dog, if you can teach these to another thing, and your mind is going to be really clear whether you know the concept well enough or not. And after you're done repeating these to another person, you'll be able to understand it even better, and you'll remember it perfectly for Monday's test. Next one is, does evolution happen in the same rate for all organisms? Right here. The answer is no, because evolution is not like making bread. It's the same process. The process is about the same. It's pretty similar, but how fast does evolution happen? It really depends. So make sure you know that. Does it usually take a short time or a long time for evolution to happen? Usually, evolution happens. It takes a long time for one species to evolve into a different species, but it usually takes a long time. But evolution can also happen within a shorter period of time. So if you can remember the pepper moth story that we talk about, that is microevolution happening within a short time, where yes, evolution is happening. There's a change in allele frequencies, but we didn't produce a new species. The next one is, what is the difference between gradualism and punctuated equilibrium? So both of these two words, gradualism and punctuated equilibrium, tells you how fast evolution happens, but there's only one that's actually correct. So if you look at this picture right here, on the left side it describes gradualism. So what it means is that evolution happens gradually, right here. Evolution happens gradually over a long period of time in a linear branching fashion. So what does that mean? We start with one species. Before it evolves into two different species, this process takes a long time, and it happens gradually. So there's a little bit of change again, and then eventually it becomes a new species after a long time. So that's why it's called gradualism, because there are changes gradually, that gradually happens. The other one is called punctuated equilibrium. Punctuated equilibrium says one species is going to stay exactly the same for a really long time, and then within a much shorter period of time, it's going to make a sudden change. So we'll jump from this look to this look. And then it's going to stay the same for a long time again, and then it's going to make a sudden change into something else, and then eventually we get to a different species. We now know that punctuated equilibrium model is more accurate to how evolution actually happens, and this is because if you were to look at fossils, you rarely see small changes in between fossils that are related. You usually see fossils that stay the same for a really long time and then changes all of a sudden. So this picture over here can also help you remember. This is a model for Gradualism, so gradualism, remember, is the wrong one. Gradualism is difference happening, there's changes happening gradually. But this is punctuated equilibrium, where big changes happen in a shorter period of time. It stays the same for a long time. Big changes happen within a short period of time. It stays the same for a long time again. So make sure you remember the difference between these two and which one is the correct model. The next one is called spontaneous generation. Spontaneous generation is a theory that living things emerge from non-living or inanimated objects. So the example given here is that if you leave meat outside, it's no longer living because you cut it off from the cow already. So it's non-living. And if you leave it there for a long time, you might see maggots growing on there, so it looks like maggots just came out of nowhere. Non-living gave rise to living objects. However, we now know that this is untrue because the maggots actually came from flies laying their eggs. And this experiment over here shows you why that's the case, because if you were to seal the container, then you don't get maggots. And if you were to use gauze covering the container, then maggots grow on top of it because the flies lay eggs on the gauze. So this is what spontaneous generation is about, and you need to know that spontaneous generation is a theory that people had previously and is proven wrong. The last one is pepper moth and bacteria antibiotic resistance. Peppered moth is before industrial evolution. The tree was white and most of the moths were the lighter colored ones. During industrial evolution, there was a lot of soot covering the tree, so then the tree became darker, and the darker moths became the ones that are prevalent in the population. After industrial evolution, the air got cleaner, so the tree went back to white because there's lichens that are white covering the trees, and then the moths, the white moths, were able to hide a whole lot better. I think it's right there. Oh wait, right here. That's a white moth. Can't even see it. So the birds can't see the white moths either. So the allele frequency of the moth population changed, and the white moths became the ones that were dominant for the population. The last part of this is antibiotic resistance. Antibiotic resistance can be developed within bacteria. So first off, antibiotic is antibiotic means bacteria. So antibiotic itself is a medicine that kills bacteria, supposedly. But some bacteria, bacteria can evolve really fast because they're unicellular and they can make offspring way faster than we do. And the result is some bacteria can go through evolution and develop, just by chance, the ability to be drug resistant, to be antibiotic resistant, meaning that they cannot be killed when you use antibiotic. So that's antibiotic resistance. The result is if you were to use antibiotic, you kill all of the bacteria that are not antibiotic resistant, so they're all dead. And then the ones that are antibiotic resistant is going to live and is going to reproduce. And antibiotic resistance, if you look over here, can also be passed on from one bacteria to another. So this bacteria over here is able to form a bridge and send this piece of red gene, the antibiotic resistant gene, to another bacteria. We call this conjugation. We'll mention it again. But conjugation allows bacteria to share genetic information with each other and give a new piece of DNA to another bacteria. And the result is more and more bacteria is going to be antibiotic resistant. And then we can't use the same antibiotic anymore. It's not going to work. Now we're on to virus and bacteria. The first question for virus is virus living or non-living? Please, please, please. It's not living. Because virus cannot live on its own, so it's non-living. Is virus a prokaryote? It is not. Virus is non-living. A prokaryote is Bacteria or Archaea. Virus is none of those, so a virus is not a prokaryote. What is virus made of? A virus has, if you can remember these two pictures, or one of these, a virus always has genetic material within the center of of the protein coat. So there's either DNA or you can have RNA as the genetic material for a virus. And a virus always has a protein coat, so it can look like this, you can look like this, it can look like other things, but there's always the genetic material, DNA or RNA, and the protein coat. Sometimes, a lot of times, virus could also have a lipid tagged onto the protein coat, and this kind of helps with the specificity of virus and its host. So virus can only infect, or one type of virus can only infect certain types of organisms, and so virus and host is very specific toward each other, and the lipid helps it. How is a cell and a virus similar? A cell and a virus is similar only because they both have DNA as a genetic material, potentially. If a virus has an RNA, then that's not similar to an actual cell. The protein is also can also be considered as a part of it, but the DNA is definitely the most important part. Can a virus live on its own? Nope. What is the protein coat of a virus called? It's called a capsid, C-A-P-S-I-D. Or what is the coat of a virus made of? It's made out of protein. What is a virus genome made out of? DNA or RNA? Is the genome of all viruses the same size? Nope. Some genomes are smaller, some are bigger, just like the genomes of animals. This next one, how does a virus reproduce? Okay, so if you look at this picture right here, a virus reproduces first by attaching itself onto the host cell, and there are specific receptors on the host cell that can match with the virus. And after this attachment, a virus kind of poops out its DNA or RNA genetic material into the bacteriophage. So it inserts the genetic material into the host genome, and then because the host has all those nucleus and ribosome and all that in order to make more DNA and more proteins, then as you can see over here, we're making more and more of the DNA copies. All right, the DNA copies are separate from the protein coat, and we're also making the protein separate from the DNA. And then you can't just have pieces of virus without a whole virus, so you also have to go through a step called assembly where the DNA of a virus and the protein of a virus are assembled together. The last step is called release, or we can call it lysis, because the virus has to slice open the bacteria or host or whatever type of host cell membrane in order to allow the release of the virus. So make sure you know that, well, I mean, you need to know all of this actually, never mind. Can a virus infect every type of organism? No, virus is specific for its host. What do we call a virus that infects a bacteria? It's called a bacteriophage. A phage is kind of like eating. Bacterial is bacteria eating, bacteria infecting. Virus is a bacteriophage. Make sure you know bacteriophage is not a bacteria. We're still talking about virus, but it's a virus that infects bacteria. So for this test, we're talking about all different kinds of organisms, so make sure you don't get confused about what these words mean. So bacteriophage, for example, might get you a little confused. Next one, does the protein coat of a virus get injected into a host cell? As you can see in this picture, the protein coat itself actually doesn't get injected. The DNA or the RNA gets into the host, and then the DNA and RNA serve as an instruction for how to make more of the DNA or RNA genome for the virus. And it also serves as an instruction on how to make more proteins, how to make the protein coat. Okay, so the protein coat itself doesn't actually get into the host. What gets injected into the host cell? DNA or RNA, or you can call it the genetic material of a virus. Make sure you know. Oh yeah, virus, a capsid, and genome are produced separately, and then they get assembled, and lysis, slice open the cell membrane to allow the release of the virus. Next one, what is the difference between a lytic cycle and a lysogenic cycle? If you can remember this picture right here, you'd be good. The lytic cycle is after the injection of the genetic material into the host, the DNA doesn't actually get incorporated into the host genome. So then what only happens is this piece of viral DNA serves as an instruction to make more viral DNA or RNA, and then produce more of the protein coat, and then you assemble the DNA with the protein coat, and lysis and viruses are made. So for the lytic cycle, the viruses are produced immediately and released immediately after the injection of the genome. However, for certain viruses, instead of only going through the lytic cycle, it can go through the lysogenic cycle. And how that works is after the viral genome gets into the cell, it becomes a part of the host genome. As you can see over here, they became one part together. So the viral genome becomes a part of the genome of the host. So as the host cell replicates and making more copies of itself and reproducing whatever, the viral genome also gets copied with the host genome, because the viral genome is basically a part of the host genome. They're together. However, if there is an environmental trigger such as heat, stress, radiation, for example, it would trigger this lysogenic cycle to then go into the lytic cycle. So during the lysogenic cycle, the virus is kind of staying dormant. The viral DNA is in your DNA, but it's not doing anything. After a trigger, this viral genome is going to start making copies of the actual virus and go into the lytic cycle and actually cause harm. Okay, a typical example is herpes or cold sore. Is caused by the herpes virus. The herpes virus is in your gene and is always there, and it will always be there if you ever had the cold sore. However, when there's an environmental trigger, this viral genome will actually start making the virus, and that's when you actually get the cold sore. So, which one allows virus to replicate immediately? That would be the lytic cycle. Which one have the virus become a part of the host genome? That's the lysogenic cycle. And the next one is a retrovirus. A retrovirus is retro because the virus itself has an RNA genome. So before the RNA can be made into more RNA copies, the RNA has to go backward and turn the RNA into a DNA. And then the DNA is going to make more of the RNA viral genome, and then proteins, and the RNA and the protein get assembled together. So the reason why we call it retrovirus is because when you learn the central dogma, the central dogma is DNA to RNA to protein. So the retrovirus is going backward. The next one, what are the two types of prokaryotes? That would be bacteria and archaea. Describe the structure of a bacteria. You can just remember this picture. A bacteria can have not a nucleus. It does have a DNA. It has ribosomes, it has a cell wall, cytoplasm, potentially a flagellum. Pillas are kind of similar to the cilia. So that's bacteria. The difference between bacteria and archaea is, well, first off, the cell wall component, what makes up the cell wall for bacteria and archaea are different. And also, bacteria can be a pathogen and actually cause diseases, and archaea cannot. Archaea is good. And archaea obviously can live in extreme environment, and archaea itself is actually more similar to eukaryotes than bacteria do. The next one is, what is a binary fission? A binary fission is how prokaryotes reproduce. So this picture right here showing you DNA makes a copy of itself, the stretches, now you have two exact copies of the same prokaryote. Okay, so it does not. Does it allow genetic recombination? The answer is no. The next one is called conjugation. A conjugation is how prokaryotes can actually share its genetic information and allow new genetic combinations. So remember when we talk about antibiotic resistance, let's say this bacteria right here has antibiotic resistance within this little plasmid. It can form a pilus, and send this plasmid, well, a copy of this plasmid into a recipient. So now this bacteria, well, this bacteria is still the same as the original bacteria. It's the same exact bacteria, except now it has a new piece of DNA. So conjugation provides genetic diversity in bacteria, which allow them to survive in new environments or to resist antibiotics, like we said before. The last one is, what are some ecological roles of prokaryotes? They can definitely be a decomposer. Remember, a decomposer is something that can break down dead things and recycle the nutrients. Producers, bacteria can actually do photosynthesis. Some of them can. Last one is nitrogen fixers, turning the nitrogen gas in the air into a different form of nitrogen so that they can be used within the body of plants and animals. So bacteria can do that. It's very important. The last one is, what's a pathogen? A pathogen is something that can cause diseases, a disease-causing cell. So that could be a virus or a bacteria. A virus, remember, is not actually a cell. The last one is, what can, I've said that 3 times, what can be a pathogen, a bacteria and a virus? This next one is protists and fungi. First off, what are protists? Protists are, here it is. Protists are autotrophic or heterotrophic eukaryotes. That is not a plant, animal, or fungi. So it's the other thing. Some protists are more similar to plants, some are more similar to animals, some are more actually similar to fungi. Most protists are unicellular. Remember that, but protists can be unicellular or multicellular. Next one, example of protists, it could be a kelp, amoeba, or paramecium. What you need to know is that a kelp is that it's kind of like a seaweed attached to the bottom of the ocean. A kelp moves through passive movement. Non-modal protists depend on water or air. So that is associated, this part on how protists move is associated with kelp. Amoeba is the video that we watched of the one with, it's kind of like a slimy thing with little legs, and it moves. So amoeba moves through using the use of pseudopods. So that's the extension of a part of the little leg in one direction. The whole body kind of follows. The last one is paramecium. Paramecium itself has cilia. So a cilia kind of looks like, if you look at the bacteria, that's not bacteria. If you look at the bacteria right here, these little hair are kind of similar to the cilia. Flagella is a long tail. You can have multiple flagella. But remember, flagella is much longer. So you might see a picture showing you a cilia or flagella and ask you, which one is it? The next one is the classification of protists. We know the classification process is still a work in process because we can't exactly put all protists in one group. The reason for that is shown on this cladogram. Some protists can be more similar to animals. So if I were to ask you which one of these protists is the most similar or the most closely related to animals, it's going to be this one. I'm not going to say this word. The next one is some protists are more similar to fungi as well. So if I were to ask you which type of protist is most similar or most closely related to this fungi, you can see this and this both come to this common ancestor. And this come to this common ancestor. They're kind of the same. I would say, I'm not going to ask you a question like that because this one's kind of ambiguous. However, it can also ask you which protist is most closely related to plants, and it's going to be this little guy, which is the Rhodophyta, if that's actually how you pronounce it. But you can also see these protists are more closely related to each other than the plants, fungi, and animals. That's why the classification of protists is still a work in process. We need to learn more about protists in order to classify a little better than this whole mess over here. The next one is, so we did all of this. Examples, all right. Next one is, what are some ecological roles of protists? How can protists cause diseases? Ecological roles are all over here. So for example, phytoplankton consists of, well, some part of phytoplankton is made out of protists, and those can feed fish and go through photosynthesis. Protists can also be a part of coral reef and produce energy for the coral. It can also provide shelter, so that's a kelp forest that can provide shelter. It can recycle waste. And the last one is pretty important, is symbiosis, where a type of protist can live inside the digestive system of termites and actually allow the termites to digest food. So the termite itself actually cannot digest food without the help of protists. The next one is what are fungi and what are their structures of fungi. Right here, they're heterotrophs. They could be unicellular or multicellular. They have a cell wall. The next one is, are they autotrophic, heterotrophic, or both? It's heterotroph. Are they multicellular, unicellular, or both? It's multicellular. What's a fruiting body? So a mushroom is a typical example of a fruiting body. A fruiting body is the reproductive organ of fungi, and it can produce spores. So spores are the reproductive cells. So people can produce gametes as our reproductive cells, and mushrooms or fruiting bodies of fungi can produce spores. So make sure you know that. The last one is what are some ecological roles of fungi? Decomposition, parasitism, can be a part of lichen as well. So that's mutualism right here. All right, the next one is introduction to plants. All right, plants, what are plants? Multicellular, eukaryotic, photosynthetic, autotrophs. That's just some words that you can use for plants. What are plant cell walls made out of? They are made out of something called cellulose, so that's a type of carbohydrate, but it's called cellulose. The next one, what type of macromolecule is sugar? Is carbohydrates. What part of a plant produces carbohydrate? What part of a plant? It will be the leaf of a plant specifically produces carbohydrate. What part of a plant cell produces carbohydrate? Then that will be chloroplast. The next one is what part of a plant cell stores carbohydrate produced? That will be the vacuole. The next one, what do plants need to survive? So all you have to do is to remember this picture: carbon dioxide, light, water, and last one is mineral. The plant needs light and carbon dioxide and water because it has to do photosynthesis. And why does it have to do photosynthesis? It's because they have to make sugar so that those sugar can be used by the plant cell in order to produce energy and allow the function, the cellular function of a plant. So that's a plant. Okay, photosynthesis. All of these are used for photosynthesis. What part of the plant receives water? What part of the plant absorbs water? That would be the roots. What part of the plant receives water? The entire plant. How was the common ancestor of plants like? They are similar to green algae, but they're not green algae. Remember that. And also, the common ancestor of all plants lived in water. Can you say green algae is the ancestor of plant? Nope. What was the biggest challenge land plants face when they first started growing on land? Will be water. What are some adaptations plant developed during evolution to allow survival of plants on land? So if it has problem getting all that water, the first thing that you can think of is a cactus. So if you think about a cactus, such as right here, think about a cactus, it can resist heat and sunlight because the heat and sunlight make it easier for a plant to lose its water, for the water to evaporate. And also the plant itself needs a mechanism to conserve water. So for a cactus, it has all these needle leaves, which really help conserving the water because it makes the water less likely to evaporate through those tiny leaves. And also plants need to be able to reproduce without water. Before plants were on land, they have to reproduce with water. So plant has to also develop the ability to reproduce without water. What are some major groups of plants? That will be right here: green algae, mosses, ferns, cones, flowering plant. So we have so make sure you understand this picture. It's going to be showing up on the test. Remember, we added a branch over here called green algae. So there's green algae, non-vascular plant, does not have a vascular system. They have to stay close to the ground because that's how they can get the water and allow water to diffuse throughout the plant, and that doesn't take very long. But in order to during plant evolution, plant eventually gained the vascular tissue, which gave rise to all three of these plants: the seedless vascular plant, gymnosperm, and angiosperm. Seedless vascular plant, you can see from the name that it doesn't have seeds and it doesn't have flowers. Gymnosperms have seeds, but it doesn't have flowers, and angiosperms have flowers and seeds. And from this cladogram, you can also see how that works, because seeds show up over here, flowers showed up over here, and then vascular tissue showed up over here. So all three of these have vascular tissue. Both of these have seeds, and only the angiosperm has flowers. Provide some examples. All right, some examples. For example, non-vascular plant can be mosses. Seedless vascular plant could be ferns. Gymnosperms can be Christmas tree, whatever that's called. I forgot what it's called. Oh, conifers. Conifers are the Christmas trees. And then the last one, angiosperm, cherry blossom, anything that blossoms will be an angiosperm. The next one, how can plants be classified based on the presence of vascular tissue? Those are our fancy words. We have the bryophytes and tracheophytes. So bryophytes are the ones without vascular tissues. So they have to stay close to the ground, live in a moist area. They're really small. They can only depend on water diffusion, and it doesn't have a xylem. So what is a xylem? A xylem does not exist for bryophytes, but it does exist for all the tracheophytes, because xylem is the one that transports water and is able to transport water for longer distance. So if you see a tree outside that's really tall, or any trees really, they all have xylem that transports water. The next one will be, what do we call, okay, we did this. Does a non-vascular plant have roots or xylem? They don't have roots, they don't have roots, and they don't have xylem. What is the function of a xylem? Transport water. What kind of environment does a bryophyte need to survive? Moist area that's shaded, potentially. What's an example of a bryophyte? That would be ferns. What else can we call a vascular plant? That would be tracheophytes, right here. What does it mean for a plant to be a vascular plant? It can transport water for a longer distance, so the plant can be taller. And then you can subdivide the vascular plant, the tracheophytes, into three categories: the seedless vascular plant, the angiosperms, and gymnosperms. So how can a seedless plant reproduce? So a seedless vascular plant, right here, such as ferns, they can reproduce by spores. So it's spores again. So the spores kind of just fall off the seedless vascular plant, the ferns, and then develop into more ferns. And then later on, we have the gymnosperm and the angiosperms. And gymnosperm depend on the seeds to reproduce, and angiosperms also develop seeds in order to reproduce. What's an example of a seedless plant? Fern. Difference between gymnosperm and angiosperm: gymnosperm is not able to produce flowers, and angiosperms can produce flowers. Examples and characteristics of each, right here, is right here. Gymnosperms can be more resistant to wind, cold, and drought. So if you think about a Christmas tree, what are some characteristics of a Christmas tree? The last one is angiosperms. They have flowers. Monocot and dicot, you don't really need to know much about that. I'm going to show you a chart on the test, and you'll get information out of the chart in order to answer some questions. But monocot, when it first develops and have its first leaf, there's only one leaf coming out of the seed. And for a dicot, when it first develops, there are two leaves. That's why it's called dicot. So in this picture, it's showing you we have xylem right here. Xylem is transporting water from the roots to the leaves and everywhere else in the plants. And phloem is transporting sugar from the leaves into everywhere else in the plant, because sugar is produced in the leaves. That's where photosynthesis is done. So we just did this one. What is the difference between diploid and haploid? A diploid is when a cell has two sets of chromosomes, and a haploid is when a cell has one set of chromosomes. when you compare gametophyte and sporophyte, which one is the haploid stage? Gametophyte, just like human gamete, is haploid. It's one N, one set of chromosomes. And sporophytes are 2N, diploid. And you need to make sure that you understand this picture right here. This picture right here. What is it talking about? A sporophyte, the reason why it's called sporophyte is because sporophytes produce spores through meiosis, and gametophytes produce gametes. So in this picture, it's showing you that a plant can first start with a zygote, and then it develop into a sporophyte, which is a diploid stage of a plant. And they can go through, the sporophyte can go through meiosis and produce spores, which is a haploid cell. Those spores can develop into gametophyte, which is the haploid stage of a plant. Gametophyte can then produce gametes, and then gametes can fertilize and produce zygote again. So you can go through this cycle again. But what you need to know is that gametophyte is the haploid stage of a plant. Sporophyte is the diploid stage of a plant. Unlike human only has a diploid stage, but plants have both the haploid and the diploid stage. The last one, what are some characteristics of a green algae? It has to live in water, mostly fresh water, and that's it. Hope this is helpful. Hopefully you'll do great on Monday's test if you put in the effort and actually watch this video. Not only watch the video, but also test yourself using the review sheet and make sure you understand and can remember all the answers. Then you'll do great. Have a good Sunday afternoon.